Hi, this is Rethik, your biology subject matter expert from MEMONEET. And in today's video, we will be concentrating on the next topic, that is meiosis. After mitosis, the next cell cycle that happens is meiosis. So what is meiosis and what are the process that comes under meiosis? That is what we are going to concentrate here. Okay. So yeah, coming to the meiosis, the process of two offspring, say like it could be the male and the female, gametes, right? So they fuse together, that is sexual reproduction. It includes the fusion of two gametes. So each will with a complete haploid set of chromosomes. Haploid is nothing but 23 set plus 23 sets of chromosomes. So 23 sets of chromosomes from the male and 23 set of 23 chromosomes from a female. Okay. So when they combine, fuse together, then it leads to what? The process actually is called as sexual reproduction and the fusion which leads to the formation of zygote. Right. So the gametes are formed from the specialized diploid cells here. And these specialized kind of cells divide their cell division that they undergo through reduction division, that is reduction of the chromosomes. Say like in human we have 46 number of chromosomes. The chromosomes will become half it is going to reduce. It means 23 chromosomes you are going to find. So whether it is an haploid or diploid, so reduction of chromosomes is haploid, right? And this is like diploid. Anyhow, so this kind of division is actually called as, so from reduction from 46 to 23, this kind of reduction division is actually called as what? Meiosis. Okay. So we come across meiosis during gametogenesis. It could be in plants as well as in animals. And this leads to the formation of what? Haploid gametes. That is, now the gamete is having a 46 set of chromosomes or what? 23 set of chromosomes, not the complete. Okay, so it is something called as reduction division. So reduction matlab haploid will have haploid gametes will be there. So that means only half of the chromosomes will be there in the gametes, so if it is related to male or female. So I'll go to the major key features of meiosis. What are the key features of the meiosis? So it involves two sequential cycles of nucleus and cell division. So one is called as meiosis 1 and meiosis 2, but only a single cycle of DNA replication will take place. So that happens in meiosis. Okay, the DNA replication and all will take place in meiosis 1. Then this meiosis 1 is initiated after the parental chromosome have replicated. So in mitosis, what you are going to observe is the parental chromosomes will replicate, right? So in synthesis phase, something you're going to see. So now the chromosomes are there. They are the parental chromosomes have replicated to produce what? Uh, an identical sister chromatides at the S phase. So that is nothing but synthesis phase. So in synthesis phase, what happens? There you will actually see the replication, synthesis of the chromosome. So you are also going to see the sister chromatides formation. So then the meiosis involves in the pairing of the homologous chromosomes and recombination between the two homologous chromosomes. So you have homologous chromosomes here. So what you are going to finally observe is you are going to observe the pairing here. Okay. So there are different two homologous chromosomes here. And ultimately you are going to observe what? The kind of pairing in any one region. Okay, so four haploid cells are formed at the end of the meiosis two. Four haploid cells in sense. So if it is you have something, two cells will actually result and give rise to four haploid cells. Yeah, we'll move to the next one. So stages of the meiosis. So majorly the meiosis has been categorized or grouped into two stages. One is meiosis 1 and meiosis 2. In meiosis, in the meiosis 1, you're going to study the prophase 1, then metaphase 1, anaphase and telophase 1. where meiosis 2, you are going to study the prophase 2, metaphase 2, anaphase 2, and telophase 2. Right. Uh, Next, we will see like what is meiosis 1. In meiosis, basically, you're going to see five category or stages of the prophase 1. So we have prophase, right? So in meiosis 1, we have prophase 1, metaphase, anaphase, telophase 1. So in prophase 1, we are going to absorb the leptotin, zygotin, pacatin, diplotin, as well as the diakinesis. So these are the five stages that you are going to observe or study in the prophase 1. So in the stage 1 of the leptotin, what you are going to observe in the figure is during the leptotin stage, the chromosome becomes gradually visible under the microscope. So you can see like the chromosomes that are visible under the microscope, and the compact of the chromosomes continue throughout the leptotin condensation, something like that. This is followed by the second stage of the prophase 1, which is actually called as after leptotin, you are going to see zygotin. So you are actually going to see the condensation of the chromosome. Because of the condensation of chromosome, so we can easily observe the chromosome under the microscope. So after the leptotin, we have what? Zygotin. Where in zygotin, the chromosome starts pairing together, and this process of association is called as synapse. So can you see there is kind of association here between the two homologous chromosome? So if this is the homologous chromosome, so there is a kind of association here. Okay. So this association is actually called as synapse, and such pairing of chromosomes. So association is there, and now that the two homologous chromosomes, they're getting paired together. So we can call them as uh, such pairing of chromosomes are actually called as homologous chromosomes. So the coloration I'm talking. So this is one. So they are same sister and they are also same chromosome. So one will be obviously homologous chromosomes. And there is a pairing between these two homologous chromosomes. So electron micrograms of the stage indicates that chromosomes synapses accompanied by the formation of a complex structure. So that is where these two homologous chromosomes, right? So if this is chromosome and this is one chromosome, so here there is an association. So that complex is actually called as synaptonemal complex. Okay. And the complex formed by the pair of the, what do you say that, synapsed homologous chromosome is actually, we call it as the bivalent or tetrad. However, these are more clearly visible in the next stage. We'll go to the next stage will be the so what happens in the packet is the first two stages of the prophase 1 are relatively short-lived when compared to the next stages that is in the packetin. So during the stage, what happens? The four chromatides of each bivalent chromosomes, they become distinct and clearly appear as a tetrad, like the way you can observe here. So in this stage, what happens is it is actually characterized by the appearance of the recombination nodules will be observed. So that means if you clearly absorb it, you'll get it. So there is a kind of recombination which is going to happen here. Okay, kind of like this. Yeah, some kind of recombination that we are going to absorb. Yeah, can you see this? So yeah, the same thing will happen here. So what happens basically is the crossing over is one thing that occurs here. Okay, the crossing over is an what do you mean by crossing over here? So it is actually a genetical exchange, a kind of genetic material being exchanged between two homologous chromosomes. Crossing over is an enzyme mediated process, and the enzymes involved is actually called as recombinase. So this is very important, my dears, because some twisted questions might uh, as correlated the enzymes which are responsible for the crossing over. Always remember crossing over is nothing but recombination. So enzyme name will be recombinase. So that is how one can remember. So crossing over will obviously leads to what? It will leads to the recombination of the genetic material. Recombination is nothing but exchange of the genetic material with the two different chromosomes. And between the homologous chromosomes is actually completed by the end of this pachytene, leaving the chromosome link at the side of crossing over. So then we have is the diplotin. So in diplotin, the beginning of the diplotin is actually recognized by the dissolution or of the synaptonemal complex. That is, what do you say that the complex of two homologous chromosomes joining together, and you will see like the recombination is complete now. So that means the crossing over is completed now. Okay, and now it is ready to get separated from each other. So this is how it is done. So here the mark that you are actually seeing, it's kind of X mark, right? X kind of mark. So that exists shaped structure that are actually called as the chiasma. So this is a position, or this is a place where actually the crossing over has happened, and now the genetic information has also exchanged. So in oocyte of some vertebrates, the diplotins can last for months or even for years. Then the last we have is the diakinesis in the prophase 1. The final stage of the meo, uh, meiotic prophase 1 is actually what? Diakinesis. So this is marked by the terminalization of the chiasma, that is separation of the chiasma formation. During this phase, the chromosomes are completely or fully condensed and the meiotic fiber is assembled to prepare the homologous chromosomes for the separation. So what happens is at the end of this formation, that is the diakinesis, you can see even though it is blue, only one part red is there. This is red, but it is one small part is blue. That means now what happened? Mutually from this, the genetic material might have exchanged here, and from this, the genetic material is exchanged here. Okay. So this is one thing that you will be observing in the diakinesis. So at the end, the nuclei disappear and the nuclear envelope also break down. The diakinesis represents what? A transition to the metaphase now. So after prophase, what you are going to observe? You're going to observe the metaphase formation. Okay. So in metaphase 1, the bivalent chromosomes align on the equatorial plate and the microtubules from the opposite poles of the spindle attaches to the kinetochore of the homologous chromosomes. So just you will be observing like in meiosis what happened, the same thing you are going to observe here. Then in anaphase 1 happens, the homologous chromosomes get separated while the sister chromatides remains associated at the end of the centromere. Then we have telophase. So the nuclear membrane and the nucleus, they again, they are going to reappear, cytokinesis followed, and it is called as diad of the cell. Although in many cases the chromosomes do undergo some dispersion and all, they do not reach the extremely extended stage of the interphase nucleus. The stage between the two meiotic division is actually called as what? Interkinase, and is uh, generally short-lived period of time. So there is no replication of the DNA will occurs in the interkinase. Interkinase is followed by the prophase 2, a much simple prophase than the prophase 1. Like prophase 1, it is complicated. It has 5 steps. But in prophase 2, you are not going to see such steps now. So this is the meiosis 2, what all the things will happen. So your prophase 2, metaphase 2, anaphase as well as telophase. It is followed by this. So prophase 2, the meiosis 2 is initiated immediately after the cytokinesis. That is, what do you mean by cytokinesis here? Yeah. Uh, the whatever the DNA they got separated, right? So usually before the chromosomes have fully elongated. Then in contrast to the meiosis 1, meiosis 2 resembles a normal mitosis. Just like mitosis, it resembles. and the nuclear membrane disappear by the end of the prophase 2. Then the chromosomes again become compact here. One thing that we need to observe here is, what do you say that? Yeah. Next in the metaphase two, same thing. Again, the chromosomes that are aligned at the equatorial and the microtubules from the opposite poles of the spindle gets attached to the kinetochore of the sister chromatids. You just need to understand where the position is and what happens and where it is attached and all. Then one thing we can do is we can easily frame the definition and we can know the answer even it is twist. The question is twisted. Then we have anaphase 2. It begins with the simultaneous splitting of the centromere of each chromosomes. The centromere is nothing but the one which is actually holding the sister chromosome, chromatides together. Along them to move towards the opposite pole of the cell by shortening of the microtubules attached to the kinetochore. So in the next, we have a telophase. So meiosis ends with the telophase 2. So this is the last phase in which the two group of the chromosomes once again gets elongated by the nuclear envelope. Then the cytokinesis follows, resulting in the formation of tetrad of cells, that is four haploid daughter cell, as you can observe here. So there has to be some significant importance of the meiosis, right? So meiosis is the mechanism by which the conservation of the specific chromosome number of each species is achieved across the generation to generation through the sexually reproducing organisms. Uh, Even though the process even though the process, it results in reduction of the chromosome number by half, then it also increases the genetic variability in the population of the organisms from one generation to the other generation. And variations are very important for the process of evolution. So this is all about the meiosis and a very beautiful explanation of this entire cell cycle. So I just made a revision video of one is what do you mean by cell cycle and the mitosis, and the second part is regarding meiosis. I hope this is clear to you all. And if you have still any doubts, you can reach me out to the comment section, and you can also reach me out to the Telegram channel. Very important thing is don't forget to subscribe, like, share with your friends, and also comments, because by your comments only I'll be getting to know whether whatever that I'm explaining is reaching to you or not. And second thing is you can actually still, if you are finding to go with some guide or some previous year question papers and all, you can still purchase the MEMONEET NTAPYQ book. So, which is a very good, it has all the previous year question papers with a well explanation part. So, yeah, this is it for today. All the best and yeah, keep watching. Thank you.